So hello and welcome everyone who joined our Intelligent Infusion Masterclass today. Just to quickly introduce myself, my name is Lea Esser, and before I started here at Cube Biotech, I finished my PhD at the University Clinics in Düsseldorf. And I joined Cube 1.5 years ago, and I'm working here in the role of a research scientist. And it's my pleasure to introduce our speaker of the day, which is Anna van Kooijen. So welcome, Anna. Just a quick reminder for people who did not join already the masterclass sessions. This is a platform we as a company created as researchers for researchers to share and promote their research with a worldwide audience. And after the talk, there will be a time for a question and answer session. And during the talk, you have the chance to type in your questions with a question mark, and afterwards they will be answered and shown on the screen. And even if later on you have further questions, you have still the chance to reach out to us via email, and your questions will be also answered soon. So now coming back to Anna's biography, Anna received her bachelor's degree in molecular, cellular, and developmental biology from the University of Colorado Boulder. And she went on to study calcium and ion channels and mitochondrial and calcium dynamics. And afterwards, she joined Dr. Chandra Thakur's lab at the University of Colorado Anschutz Medical Campus, where she is currently completing her PhD. Her research in general focuses on designing and implementing novel optogenetic and chemogenetic tools to probe and manipulate protein function in cells. And her major goal is to remain familiar tools as modular building blocks. Within her talk today, she will show us how she proved that Strep-Tactin and the Strep-TagII that are originally designed for protein purification and biochemical assays proved to be excellent modular components for creating a chemically controlled tool in living cells. And yeah, so we are really looking forward to your talk today, Anna, with the title Strep-Tactin and Strep-TagII Reimagined, a modular, genetically encoded approach controlling protein function inside cells. And with this, I want to thank everyone for joining today. And yeah, the stage is yours, Anna, if there are no further questions.
All right. So again, I'm really honored to be here. Especially since I think this may be a little outside what is normally talked about here in these master classes by Cube Biosciences. So let's jump into it. Okay, so we are going to start by reviewing the central dogma of molecular biology. So as you all probably know, in the cell, DNA is transcribed into RNA, which is then translated into a functional protein. Now, let's say that we as scientists wish to turn on or off some protein's function in the cell. So we have a couple methods to do this. At the DNA level, we could knock in or out a new gene using CRISPR or Cre locks. Or at the RNA level, we can use siRNAs to interfere with our RNA transcripts and knock down that gene. Or at the protein level, we can find some drug that binds to our protein to either activate or inhibit it. Now, these are all really great methods, and obviously they have led to some incredible scientific discoveries, but they can have a few drawbacks. Like CRISPR or Cre locks and siRNA methods are labor intensive, and they can take a long time, which allows cells a chance to compensate for these knockdowns or knockouts. And then to design a drug to inhibit a protein could take you just years and years of research and development. And if anybody here works in drug development, they know that that is no small feat to find a new drug to affect a protein. So in our lab, we ask, what if we could acutely control that protein function with an experimenter-defined input? So that brings me to the specialty of our Tucker lab here, where we work to design genetically encoded synthetic switches. So here are just two examples of what these could potentially look like. So in both cases, we have the same effect. We have some protein domain that clusters in response to an experimenter-defined input. So this one on the left clusters in response to light, and the one on the right clusters in response to drug. We have used, and others in our field have used these domains, and we've attached them to a lot of different proteins of interest to affect their function. But what I'm talking about today is a completely different class of switch. And I think all of the biochemists with me today might be a little bit familiar with the pieces that we use for this one. So Strep-Tactin, Strep-Tag, and biotin. These three obviously have been used for years and years for protein purification, and like thousands of proteins have been purified using Strep-Tactin. So typically a recombinant protein will have a Strep-Tag2 attached to us. This will bind to a Strep-Tactin tetramer. Then when we add small molecule biotin, the Strep-Tactin will preferentially bind to that biotin and release the Strep-Tag2. Now, this is actually a perfect candidate for one of our in-cell synthetic switches because we have a non-toxic user-defined input with biotin. And at only 6 amino acids, that Strep-Tag2 is minimally invasive and relatively inert. And so we can insert it into several different places within the protein of interest without affecting that protein's function too much. So how do we plan to use this to control protein function? Well, here was our schematic for our idea for this. So I want you to imagine two proteins that normally interact in the cell, seen here in this pink and purple color. We fuse our Strep-Tag2 on or near the interacting interface, and the Strep-Tag2 is small enough that theoretically it will not interfere with this protein-protein interaction. However, this tag has an affinity for our bulky protein that can act as a steric blocker. Here it's Strep-Tactin, obviously. When our Strep-Tactin steric blocker binds, it'll prevent interaction between these two proteins. Then adding a small molecule chemical inhibitor, biotin, that preferentially binds to that Strep-Tactin will release this block and restore the protein-protein interaction. So this is the general concept of this small molecule induced tool. We first wanted to characterize the Strep-Tactin and Strep-Tag interaction in the cell. So to do this, we use one of those clustering tools that I introduced earlier. This is a light-induced tool called Cry2-olig. And in response to blue light, we should shine just like a blue light on the cells. This protein will form these oligomers in the cell. And we can see these visible puncta form very quickly after exposing our cells to light. So what we do with this is we add a Strep-Tag2 to the Cry2-olig. Then we co-express this in the cells with Strep-Tactin. So if these two are interacting in the cell, we will see the Strep-tagged Cry2-olig pull that Strep-Tactin into clusters along with it. And so we think about this as a in-cell co-IP, essentially, that we can visualize at a very fast time scale because we're looking at it with a microscope. So as you can see here in these cells, soon after light exposure, this Strep-tagged Cry2-olig and Strep-Tactin are co-localized into these clusters. Then Strep-Tactin is released from these clusters about 5 minutes after we add that biotin. This process is actually very quick. It happens in under a minute. And so that tells us, with this fast time scale, that this is a really effective tool to use in cells. It works really quickly right after we add that biotin. So we think that this would be a good candidate for downstream applications because of that. Now, one thing that I haven't mentioned yet about engineering these tools is that to have a more effective switch, you usually want them to be reversible. And this makes a lot of sense, right? Like if you want to turn a switch on, you're going to want to turn it off again eventually. And we worry that this might be a little bit of a challenge for the tool because if you know a lot about avidins from which Strep-Tactin is which Strep-Tactin is derived and biotin, so avidin and biotin interaction, you may know that they bind really, really tightly. And I wasn't sure that I would be able to overcome that to reverse the tool, that really tight biotin and Strep-Tactin interaction. However, we wanted to give this a go anyway. So we thought maybe if we add free avidins to our cell media, it could potentially act as a biotin sponge because it has a slightly higher affinity for that biotin than our Strep-Tactin. So yes, we would add free avidins to our cell media. Essentially, it would act as a sponge to pull the biotin off the Strep-Tactin and allow it to rebind to that Strep-TagII. And honestly, this was very surprising to us, but it worked quite well. So as you can see, after adding biotin, the Strep-Tactin is released from clusters. Then when we add free avidins here at this little red arrow, you can see the Strep-Tactin slowly returns to our clusters. So this was a really exciting result for us and tells us that this is a pretty effective tool for an engineered chemogenetic switch. Now, obviously, the point of designing these is to be able to control some protein-protein interaction in the cell. So I'll remind you of my original schematic here. And we sought to find some enzyme-substrate interaction that we could apply this to, now that we know how well it works. Okay. The first thing that we thought to use was TEV protease. So here I show how we used our tool with TEV protease. Essentially, we designed a reporter. It was a GFP connected to a GST via TEV cut site, flanked by a Strep-TagII on either side of the TEV cut site. So when Strep-Tactin is bound, the TEV cut site is essentially protected from proteolysis. The Strep-Tactin tetramer forms this cage that prevents that TEV protease from accessing that cut site. However, adding biotin exposes that cut site and allows for TEV to access it and subsequently cleave it. Go back. So I'll remind you, this process is happening in the cells, and then we measure that reporter cleavage by lysing our cells and using Western blot to probe for a band shift. So as you can see here, with Strep-Tactin bound to the reporter, we get a significant protection from cleavage. You can see here on our Western blot, this top band would be our uncleaved band, and this bottom band is our cleaved band. So that Strep-Tactin binding causes a big band shift towards the uncleaved band level here. And then adding biotin reverses that protection and allows for cleavage to occur again. So one of the benefits of these tools is that you have this user-defined start point. So you can say, I'm adding something and I want enzymes to begin at that point. This allows us to monitor our kinetics in cells because we have that specific start point. So as seen here, we are able to do a time course and monitor how quickly this happens in our cells. We also did a biotin titration so that we could understand the dose response to biotin. And we get significant cleavage after adding about 10% of our, or 10 micromolar biotin to our cell culture media. Now I would like to point out, I'm sure you noticed our reporter has a cut site flanked by 2 strep tags. This actually allows for an expanded dynamic range because the dual strep tag constitutes an avidity effect. So having 2 strep tag here allows that tetramer to bind really tightly to our binding sites and get really effective protection. This was not as effective if we only had a strep tag on the N or C terminal of the reporter of our cut sites. So that was really interesting. So this was all great, but like I said, we like these tools to be modular. We like them to apply to a lot of different things in the cell, and we like to demonstrate that it's not really just a one-trick pony, so to speak. Like it's something that we can take these pieces and put them on a lot of different things. So we wanted to try something completely different from a protease. And we landed on gene transcription. So here is another schematic of how we applied that Strep-Tactin, Strep-Tag, and biotin-2 to control gene expression. So here we have a GAL4 binding domain connected to a VP16 transcriptional activator domain that is flanked by 2 Strep-TagIIs. So typically, as you can see here on the bottom right, the GAL4 will bind to our reporter gene, and then the VP16 will recruit transcription machinery and activate that gene expression. However, when our Strep-Tactin is bound, it will cage that VP16 and prevent reporter gene expression because that VP16 can't recruit that transcription machinery to the DNA. And then, of course, we can activate that gene expression again by adding biotin to our cell media. So we tested this with 2 different reporter genes here just to see how well it worked. First, on the top right here, in these blue cells, you can see we used a GFP reporter. So first, we have our positive control. This is just a completely unblocked reporter. No Strep-Tactin is added. And then we have our blocked condition. As you can see here, there's very low background when the VP16 is blocked. Then after 3 or 24 hours, we see significant increases in the amounts of GFP expressed in the cell. Then to get a more quantitative look at this, seen at the bottom right here, we also used a luciferase reporter. Again, we began to see significant expression after about 3 hours, and even more at our 16 and 24-hour time points. As you can see, this worked actually really quite beautifully. And honestly, our biotin-induced gene expression works even better than that biotin-induced proteases, because we saw this incredibly low background in the strep blocked condition prior to adding that biotin. And we thought, wow, that's really exciting. Like, maybe because of this low background, perhaps this would be a good tool to use for activating transcription of a gene that could be really highly toxic to the cells. So here we use MLKL as a reporter. MLKL induces necroptosis, which is a type of cell death. So essentially what we're doing is we're using the system to induce expression of a gene that will kill the cell. So we don't want any basal levels of this because the cells will start to die. They're very sensitive to it. Here is our negative control. So our cells are transfected with only the reporter gene and not the transcriptional activator. Then we add that transcriptional activator. So this is our positive control, essentially. We obviously see tons of cells die here between around 15 and 20 hours after transfection. And of course, when we block it, we see very, very low cell death, telling us the system has a really low background. And then again, we add biotin. You can see where we added biotin is denoted by this red arrow to induce that gene expression that causes our cells to begin to die. And you may look at this and notice that our blocked condition seems to have even lower background than our negative control. And we think that this may be because that Strep-Tactin block prevents even those really, really low levels of random transcription of this reporter gene in the cell, even when you don't have that activating domain bound to it. So it essentially acts as a transcriptional repressor, and then we can very quickly activate it when we add that biotin. Okay, so thank you so much again for having me here. In conclusion, Strep-Tactin and Strep-Tag, they're good for more than just protein purification. And I hope that this can inspire everybody to have a little bit of a creative look at their tools that they might use every day with a little bit more of a creative eye, because you can really repurpose a lot of stuff for all kinds of different things. For example, we repurposed the Strep-Tactin and Strep-TagII to create this modular inducible tool. And this tool is really versatile. Like we showed in this experiment that we used it for both protease, so substrate enzyme activity interaction, and also we used it to induce gene expression. And then we have a couple other things we're working on in the lab too, where it was we're finding a couple other uses for it currently in the lab. Just a couple quick acknowledgements. Thank you to everybody in my lab. My PI, Chandra Tucker, obviously has been very helpful, and then everybody else who's been supportive, and our funding. So with that, I would love to take a couple questions, and it looks like I have some ready to go here.
So yeah, Rebecca, and thank you for this really great talk. It's really interesting to see how flexible the use can be of this tech, and it's amazing how good your technology works. So really incredible.
Thank you. All right. So should I go into these questions now?
Yeah, we're starting now. So your first question will be, do you find that natural biotin in the cells or in the cell media causes background activation of the switch?
So, if you remember with that protease experiment, we were able to do a biotin titration. The natural amount of biotin in the cell is around 10 nanomolar, I think, in cell serum. So it's pretty low levels. And what we found is that level of biotin is below the necessary concentration of biotin needed to activate this switch. So we haven't found that to be a problem. And any background we have is typically because of transfection differences from cell to cell, because it's hard to control how much of each piece of the tool is being expressed in these cells.
So the next question is then, I believe everything you have shown us was done in cell. Have you tested and characterized this tool in vitro?
Yes. So we did test this out with purified components, and we found that it works very well in vitro. Honestly, a little bit better because, as I said, transfecting the cells, they can't. We don't have a ton of control. The cell is an organic bioreactor, essentially. So we don't really have a lot of control about how much levels of each of these pieces is expressed in the cell. When we do this in vitro, we have a lot tighter control over that. So we can actually reduce that background for that protease, something like that protease experiment, even more.
So the next question is, did I get it right that regeneration via the biotin sponge only regenerated Strep-Tactin not fully, but only to a certain degree?
Yeah, we don't think it can regenerate fully. I mean, a lot of times these tools, you switch them on and off again and you start to see a reduced function. It doesn't regenerate it fully, but we think that it may be able to regenerate it enough to be effective for a couple rounds of turning it on and off. But it kind of depends on what you're using it for. You might want to, and the cell continues to, transcribe the pieces, so it'll continue to transcribe the Strep-Tactin in the cell. So hopefully, if you were trying to do this over a very long time course, we would see an increased recovery. But yeah, we don't get that full recovery with our biotin sponge washout. So.
And the second question, if you would allow, how does the gene activation with Strep-Tactin and Strep-TagII compare to other commercially available tools?
So what we've seen, TET has been really highly optimized, and it's been used by tetracycline and doxycycline TET on and TET off systems. These have been very highly optimized to activate gene expression. This one is, we're just finding it out. So, but with our initial experiments, we find that we are able to get a similar dynamic range to that TET-on system. So even without just like years and years of R&D and optimization, we're actually able to achieve something that's quite comparable. And we find the biotin is like a really non-toxic, and it's easy to supplement, even the body with biotin by just taking an oral supplement or an intravenous supplement of biotin. So that could potentially be an interesting way to activate a gene in the cells.
Yeah, maybe I have to say I worked a lot with cell culture, so induced with tetracycline or doxycycline, and I even have the feeling now that I saw your data that it's just that the uninduced controls for tetracycline or doxycycline controlled cells are even worse. So worse than what you showed, so that your switch is even more effective, at least the off rate.
We were so surprised to see how effective it was, and we think that Strep-Tactin is we think it acts as because it's so bulky, it prevents any like low background, and we found that to be really exciting. So I think it was it and once you unblock it with the biotin, you already have everything present and ready to activate the gene expression. Like everything is already in the same spot. So
yeah, that's really cool.
It's pretty cool. Yeah. So we get, compared to our positive control, we actually get a much steeper slope of activation after adding biotin and unblocking it. So it was really interesting. Yeah.
So I think these were our questions for now. But as I said in the beginning, if further questions will come up, we will reach out to you via email. So then thank everyone for joining. And of course, you, Anna, for giving this great talk today.
Yeah, thank you so much for having me. It's been so great to talk with you all, get to know you.
So then see you. Bye.
Bye.